Next we have Dr. Alejandra Solache, who's Head of Product Development at Abcam, and going to tell us all about some of the exciting work that we've been doing there on knockout validation. Alejandra, over to you.
Thank you, Jonathan. So I want to start today by asking how many hours of your careers have you wasted working with antibodies that don't work as they should? I mean, if we added them all up, probably you could have finished your PhD 6 months earlier, or actually advanced to be able to publish a lot earlier and not being scooped by somebody else. So I think this is the other aspect of antibody validation that we are not recognizing, how this is actually impacting not just a huge amount of cost, careers, and reproducibility, obviously. And at Abcam, it's part of our mission to help scientists discover more faster, and we want to do this by, we know that we have a responsibility to produce and to provide you with the products and reagents that work as they should the first time. And this is basically what I want to talk to you about, a couple of the initiatives we have to do this. I'm not going to repeat this because everybody is already, that's the reason we are here. Reproducibility is a major issue in science, and is due to, in many ways, people publishing selective results, lack of proper controls, but in a big part of it is lack of reagent validation. And you must wonder, why is it now becoming more prevalent, since, I mean, we have all been interested in, science has been focusing on reproducibility and reagent validation for the longest time. And we believe that one of the reasons that we are seeing this more often is really with the advancement of science, and the, now we have access to much better methods to test and the specificity of our reagents. So there are many new tools that now being applied to antibody validation, for example, mass spec. We are able to knock down proteins in cells at a much higher rate, and these technologies are becoming widely available. So researchers are able to actually test these products in a much better way and much more precise than ever before. So this is probably one of the reasons that we are seeing this increase in potentially retractions. And we believe that antibody suppliers have a massive responsibility here to improve consistency, specificity, and particularly transparency. And I mean, as we have discussed before, these are things that, that really are going to make a big difference if we all take care of this. And I want to discuss with you some of the things we are doing at Abcam to improve consistency, specificity, and transparency. In terms of consistency, really is focusing on generating, in particular, all reagents at high standards and adopting high standards manufacturing practices. And for example, in the case of an antibody, in order to produce a good antibody, there are many, many things that go into place. You have to really dedicate a good amount of time and thinking and to the antigen design, because rubbish in, rubbish out, basically. And also you have to have a really good immunogen production, or if it is a peptide, a trustworthy peptide synthesis. And every step of the way needs to be properly quality controlled. And obviously, once you have the antibody, you have to take really good care that the specificity and the qualifications of that antibody are the same every lot. Then moving on to specificity, obviously testing in the right applications and using appropriate controls, but also really moving on to utilizing the most appropriate applications as they are evolving and being discovered. And this is something that I will be focusing on more today, our initiative to validate our antibodies in knockout models. And this is actually very exciting. It's the first time that we are talking about this publicly, and we just are marking a milestone of publishing 500 antibodies that have this stamp validation with knockout. So the last part really is transparency. I'm not going to focus too much about this, but obviously this is hugely important. And as Jonathan mentioned, in Abcam, we have strived to be very transparent, and we probably have been one of the most transparent companies in terms of data, because we have, since the beginning of Abcam, been asking people to provide their data, and we publish their data, positive or negative, with the particular antibody. And this has helped us really identify issues so that we can remove antibodies from the catalog when they don't work, or actually optimize them and introduce better quality controls when they work, and just make them better. So what are we doing in terms of consistency and manufacturing? So I'm not going to go into detail with this. You already all know about the differences between polyclonals and monoclonals. Polyclonal antibodies have a place in research, and they have been utilized for many, many years for some applications, because their nature of binding multiple epitopes in the same protein allows them, for example, to be quite good for immunoprecipitation or CHIP. But obviously, they are a little bit more tricky and annoying in terms of keeping quality control right. Monoclonals are obviously much better, and if you keep control of your manufacturing practices, they are quite stable. Obviously, when this is not taken care, you probably could have the epitope being lost, and you stop, the antibody may stop the production, the hybridoma may stop the production of the antibody. Obviously, that is something that you can prevent by having a recombinant antibody. And we are striving to produce all of our monoclonal antibodies now through recombinant technology. And in this case, we have actually now most of our rabbit monoclonals, 90% of our rabbit monoclonals are actually recombinant. And so we have right now around 9,000 recombinant rabbit monoclonals. And we are increasing this by, as you probably know, we recently acquired Axiomics technologies, and this is going to allow us to really go much faster in our development and discovery of recombinant monoclonals. Not necessarily rabbit, but we cannot do any species here. So regardless of if the antibody is a polyclonal, monoclonal, or recombinant, validation has to be very stringent. And really, this is something that we need to remind ourselves. Obviously, for polyclonals, you have to do even more. And monoclonals, you can trust that if the clone is kept consistent, potentially you can carry out some of the applications and believe them that they are going to be behaving the same, but still you will need to be testing batch to batch. And so here, I'll just start talking to you about what we are doing in terms of validation. And as I mentioned earlier, antibody validation has been evolving since antibodies were first discovered. And when we had access to some applications for some particular antibodies a long time ago, and we are constantly reviewing our catalog and improving it and adding different pieces of application. But in general, most of our antibodies are required to have several types of applications. So we think of antibody development and antibody validation as a holistic view, because you really need to see that the antibody is performing well in multiple applications and not just in one. So some of the new applications, actually, this is an example that I have in terms of what do we do or what we have done. So this is the history of Ki-67. It's a polyclonal antibody that we released in 2007. And so the original test for this was in immunocytochemistry in human tissues. We thought, well, this is actually, in a few months later, we added immunocytochemistry in mouse. Since this is a proliferation marker, and it's also cancer biomarker because of that, people really needed to use this in IHC. And some of our reviews, app reviews, were showing that the antibodies were very good in IHC. But we wanted to make sure that every single lot was going to be working in IHC. So then we incorporated IHC validation in both human and mouse. And we found that only 66% of the batches we had available were able to recognize the antibody in IHC, human and mouse. And so we removed the other batches, and we kept only the batches that are able to recognize these different applications. So this is an example of how our validation is evolving constantly over the time. So now we are actually using novel technologies to better assess reagent specificity. In cases where we know the antibody immunoprecipitates, we can use IP, mass spectrometry. We have been using also protein and peptide arrays, and we have been using also knockouts or knockdowns or knockdown model, particularly in cases where, for example, the knockout is not a viable, will result in not a viable cell. That's a good method to use instead. So this is an example of the use of peptide arrays. For example, we use peptide arrays to validate and to help test the specificity our histone of our antibodies to histone-modified peptides. And we have developed a peptide array that contains over 400 peptides, and each peptide is spotted at 6 different concentrations, and we do this in triplicate. And we compare the specific binding of to the target peptide versus off-target peptides. And we believe that this is quite a robust method to actually decide at the very beginning of the development also if the protein, if the antibody is going to be specific or not, and we can stop the development if we see that this is not working. So now, what we are doing in terms of knockout models. So as you probably are aware, knockout models are basically recognized as a true negative control by many people, and many people consider them the gold standard. I personally don't think there is a gold standard for antibody validation. There is a specific high standard depending on the target. So knockout models are a fantastic way for us to test specificity. And so we have been doing a lot of these in the past, collaborating with people or doing small partnerships. And this, for example, a beautiful data of a collaboration we had by testing to test the anti-PD-L1 antibodies. And we really wanted to get into a much higher throughput to be able to test our antibodies in knockout. And we were very excited when we started thinking we could potentially use CRISPR-Cas9 to do knockouts, and we could develop our knockouts cell lines ourselves. And we thought, well, yes, this will be great, but it's going to take us forever. First of all, we will have to develop all these cell lines, and it's not going to be very efficient. So we kept thinking, what can we do about this? And well, we thought we need to collaborate with somebody that already has the cell lines. And we actually started an exclusive supply agreement with Iceon Discovery, who had already thousands of cell lines knocked out. And the beauty of their system is that their cell lines are utilizing the CRISPR-Cas9 technology, but they are actually haploid cell lines. So this means that when you have, when you do editing of the gene of interest, you are editing only one single copy of the gene, and you won't have the masking effects that you could have on a diploid cell line. So this was a really great thing for us, because with this technology, we now have access to a much larger number of cell lines that we can utilize, and really we can go at a much faster pace. So how are we doing this? We are testing the wild-type HAP cell line, the knocked out HAP cell line, and we add our additional control cell lines where the target of interest is endogenously expressed. And what we will do these tests either by immunocytochemistry or Western blot, depending on where the antibody is supposed to be working. What do we expect? We really expect kind of the good, the bad, and the ugly, because as we know, that's how antibodies are. We would expect the ones that are specific, the ones that have some off-target binding, but actually have recognition, specific recognition of that band of interest, and the ones that are really ugly. So specific for the target of interest, we have the band of interest gone away in the knockout, and we have the antibodies that recognize the protein target, but still will have some off-target recognition and of other proteins. And actually, we didn't need a knockout to tell us that we had off-target recognition, because we could have seen that in the endogenous cell lines. And then we have the antibody that really is quite bad, and we hadn't been able to recognize this because it was expressed. We could only see a positive cell line there. If we have had a potentially a negative cell line, we could have identified this, but we missed it. So these products are then removed from the catalog after careful evaluation, because there could be cases where the target recognizes an epitope that is non-mutated in the knockout cell line. So we have to be careful with that. So going back to our story of Ki-67, basically, after we have added all of that validation and batch-specific testing, now that we have access to the knockout validation, now we are adding knockout validation. In this particular case, we found that 94% of our batches actually were positive or were specific. So we were very pleased with this, and we were very pleased that we could actually remove a couple of batches that were not specific in the knockout. So this is a really big milestone for us, and we have now completed 500 knockout validated antibodies and against 100 targets, and we are now working on the next 500. As this has been mentioned before, we believe this is something we need to do as a collaborative effort in terms of improving reproducibility in science. Suppliers have a huge responsibility to produce the reagents in the best way possible with the highest standards. And we need to also collaborate with researchers for them to, again, as it is being mentioned already, use proper controls, share your data, publish with detailed information, and obviously journals are already enforcing that all of this information is in place. And we are driving higher industry standards for antibody validation, and we believe that this is our way to contribute to reproducibility of scientific research. And before I take any questions, I would really also like to hear some suggestions from you of how you think we can collaborate to improve this situation.
Thank you, Alejandra. Before we take some questions, I got asked at a conference, and somebody said, well, how do you feel when you find out that Abcam has supplied a non-specific antibody? And I replied quite truthfully, and I said, well, it feels as though a little part of me dies. And then some bright wag in the audience called out, why aren't you dead then? But I mean, I think, you know, the reason I'm not dead is because of the efforts that we're putting in, which makes me immensely proud, and the success stories that come out of that when I hear about the projects that are enabled by our antibodies, which obviously vastly outweigh the ones where, you know, we do inevitably supply the antibody that's not specific. But hopefully we go out of our way to then make sure that we put it right, make sure everybody who's had that antibody, you know, gets notified, is recalled, replacements offered, etc. So I would like to open up for questions, comments, and feedback.
Hi, I'm Carly Dix from AstraZeneca. I think the effort that you're making with the knockouts is fantastic for the antibody community. I was just wondering, with your HAP1 cell lines, if you have problems with your targets not being expressed in one particular cell line that you're using, and how you're going to deal with that.
Yes, so there will be some targets that are not expressed on that. Then in that case, we will go to a diploid cell line that we have knocked out, either we obtained it from Horizon or we developed it ourselves. And we are working with other partners too.
Okay, I was just wondering what proportion of targets you would estimate you would have that problem for, whether, you know, the HAP1 is a particularly good strategy for most targets.
It is for a huge amount of targets, actually. I don't remember, do you remember how many?
12,000 proteins expressed.
Yeah, it's covering 12,000.
The protein. Yeah. Actually, one of the things I remember when I was doing my work on BRCA2, the best source of tissue that expressed everything was day 9 mouse embryo. But clearly you can't mash up mouse embryos for thousands of antibodies. But they seem to just, that day 9 mouse embryo, for some reason, I don't know why, developmental biologists would probably know better than I do, but that just seemed to switch on everything. But no, these haploid cell lines don't.
I was wondering whether, alongside those studies that you're doing with the CRISPR knockout cell line, you would do another round of validation by identifying the target by IP mass spec, just to check whether actually the knockout is not targeting a related protein that could also be downregulated in its expression, and your antibody could non-specifically bind to that protein.
I'm really not.
Oh, sorry. I was wondering whether, alongside the validation using the CRISPR knockout cell line, you would also do some IP mass spec studies as a confirmation that the antibody is really specific to that target and not another protein that could be downregulated.
So we do control the experiment by using multiple antibodies to the target. So that's one way we are controlling the experiment. But also, yes, in some instances, we want to go a step further and see what is actually the specificity of that antibody, even if they show not specific at all. It could be that they are recognizing family members or isoforms, something. So in those cases, if the antibody is utilized by many people, then we are trying to go to do an extra step, either mass spec, if it's antibody that IPs, or protein arrays, for example, or any further steps. So that's something we are starting to do, yeah.
Hi, I'm Chris Ferguson from PLOS. I was interested in the transparency aspect of your presentation and how you say that and how that ties in with your removing antibodies that you can't validate from the catalogue, you said. So are you publishing this somewhere, saying that this is removed for this reason? I ask because in publishing there's quite a negative stigma to retractions, and
Yes.
actually we think differently. We think it's a valid record of science and is important
Yes.
to share. So
Absolutely, I think that's key, and I think that's part of our responsibility. So what we do when the antibody doesn't work is we notify the customers that have purchased that one, so that if they feel like they need to do that retraction, the problem is we don't know specifically what protein recognizes at this point. But we leave the data sheet in the website, even though we remove the antibody, because we want people to go back and see why that antibody is not available anymore. And we put the data for the knockout there.
Great. I think we've got time for a couple more. We'll take one over here, please, and then we'll take
Yeah, I can just yell. How are you deciding which one to test for knockout?
So obviously, with this strategy, with the hub cell lines, we have a limited universe, the ones that are expressed in the hub cell line right now, and also the cell lines that are available from Horizon. So that is
So
the
you mentioned
first.
a couple thousand. How are you
Yes.
choosing which of those couple thousand?
It's based on what we believe our scientists need the most. So we are kind of going on a criteria of what is more important for people, and hearing the feedback as well from customers. We talk to thousands of scientists from our scientific support team, and they tell us what kind of targets they want to see validated.
Okay, and then from the 9,000 lab maps, have most of those been converted recombinants from hybridomas? From the
Yes.
original epitomics hybridomas?
So 9,000 are actually recombinant
And they've been converted from hybridomas?
from hybridomas, yeah.
Okay,
So really beautiful work on the knockout stuff. So again, I'd like to come back to the transparency issue.
Yeah.
So the ultimate transparency is that you publish the sequences of your recombinant antibodies.
Mm-hmm.
And I know that Mike Weiner, the CEO of Axiomics, was a proponent of that. And I know that the industry response to this is we can't do that because then we lose our IP and people will be copying us. I'd actually like to hear your response to that and argue that, in fact, the barriers to entry to producing high-quality antibodies are rather high. And I think publishing sequences is not such a danger to your IP. And also that the problems that the antibody market suffers are far greater than other competitors copying antibodies. You've got, as Jan Bruskill behind me has said, you've got multiple reselling between vendors and primary providers. You've got batches that just change in midstream. People like to keep the same lot number because they know that if they change the lot number, something like 30-40% of scientists will stop purchasing. So one way to really fix this is a matter of publishing the sequences of the antibodies. Scientifically, I think it's very difficult. I don't think anybody from the industry has scientifically disagreed with this. All the arguments against it are all commercial.
I think there may be a point that the industry moves to that. I'm not sure we are quite ready for that. I think the problem is the cost that will be involved into moving to that. And unfortunately, I mean, the economics of producing antibodies has to play has something to play here. And then you will have to get a lot of IP protection and stuff like that, and possibly the cost of these reagents could go higher. So I don't have the right answer. I think it is, maybe in the future we will go to that point, but I am not sure that we are quite ready for
I mean,
that.
you have 9,000 recombinant antibodies, the sequences of which you have, so to actually publish those would not require very much work. You'd just add an extra line to your data sheets.
It will just require IP to protect that.
Well, we know that you can't protect antibodies for research reagents. It's too expensive. And even if you could, you would never fight to against an
So-
infringer because the cost of lawsuits is just too high.
But you know, to be honest, I think that the issue is then somebody could start producing that sequence. They don't. Any antibody any antibody company in a garage could start producing antibodies with your sequence.
There is a precedent for this. So when we first started Abcam, there was some demand to know what the peptide sequence is. So sometimes the scientists say, can you release the peptide sequence? And we started to do that, and we very soon found out that competition just said, oh, thank you very much, and they'd put an equivalent antibody using our proprietary peptide sequence that we'd put a lot of work into. So then we ended up saying sort of a compromise, because it was really difficult, because it was, well, hang on a minute, if we publish the peptide sequence, we know we're going to get ripped off. So we had a compromise, which is that we would say, if you require the peptide sequence for this antibody, please contact us, and then we could go, you know, are you legitimate or not, or are you just going to make the antibody and you're a competitor? And that's how we got around it. But I'm on your side here. I think that, you know, to do it would be wonderful. Commercially, it's really, really tricky to do. We've had our fingers burnt, as I said before, with the peptide sequences.